Howdy, Eggs! I want to welcome you to the inaugural lecture for the first-year program in chemistry. This series is intended to bridge the curriculum that you are getting in class and the real world. The intent is for us to prove that general chemistry is not only interesting, right, but also very important. Before I introduce the speaker, I need to thank a number of people. First, the staff at Rudder Theater and the folks from KMU, our local PBS station, who are here tonight to record this lecture. The lecture itself is supported by the College of Science and the Department of Chemistry, as well as the Center for Teaching Excellence here at Texas A&M University. Special thanks go to Thompson Learning, who have underwritten this first lecture. Our speaker today is a professor of radiology from UT Southwestern Medical Center in Dallas, one of the gems. Okay, so as a policy, you know, we gave UT one species, the species that can complain and sue, and we recognize as one of the premier veterinary schools in the country that we're responsible for all the others, right? All right, so Dr. Mason, who comes to us from UT Southwestern, took his PhD in chemistry in 1986 from a school called Cambridge. He joined Southwestern later that year. He's now a Texan, and we're really glad to have him. He's a recognized world leader in diagnostic medical imaging. Statistically, half of the people in this room are going to come down with cancer, and most of us will succumb to it. It's folks like Dr. Mason who are doing the research and clinical aspects that should prolong all of our lifetimes. He is a recognized leader in diagnostic imaging. He's a member of the Royal Society, the American Association for Cancer Research, the International Society of Magnetic Resonance Imaging in Medicine, and the Radiation Research Society. In 2004, he was honored as a chartered scientist as a member of the Royal Society in Chemistry and the European Union. We're glad to have him. Dr. Mason, would you please come forward and give us your lecture entitled Diagnosis and Treatment of Cancer with General Chemistry, Innovations or a Role for Innovations in Imaging. Thank you.
Thank you very much, Dr. Schmarek, for that kind introduction. It's certainly a great honor and delight to be here to give the inaugural lecture. It's also very exciting to see so many people interested in the sciences and chemistry in particular. As you heard, my original training was in natural sciences, and then I went on to specialize in chemistry, but for the past 20 years, I've been applying that to biomedical problems, particularly imaging related to cancer, and how can we move from current practice to the future whereby we may be able to eradicate cancer, improve the outlook for each of you. Let me start by showing you some members of our team. Nowadays we don't work in isolation, but rather as large research teams. A number of these are my students, young faculty, mentors. This was at a molecular medicine meeting in Houston last year. But the question is, if you look at these individuals, how could you tell if any of them had cancer, any of them had a tumor? Fortunately, due to innovations over the past several years, there are clinical modalities to look inside the body. The most popular, the most applicable and least invasive is magnetic resonance imaging. This is a clinical instrument, operates at 1.5 Tesla, because the magnetic field inside there is about 1,000 times what you have in the Earth's magnetic field. Magnetic resonance, it's relatively new. Just 50 years ago, the Nobel Prize was awarded for the discoveries in physics. Following that, it was really adopted by chemistry. It was ideal for doing structural elucidation. But most recently, it's been embraced by the biomedical community and radiologists because it can look inside the body. Now, as I mentioned, our initial instrument, this one, is a 1.5 Tesla, but we've just ordered our institution new instruments that will have twice and essentially 5 times the magnetic field. This gives better resolution in terms of space and much better indication of the chemistry that's going on inside the body. What does MRI do? One of the things I wanted to introduce was initially a simplistic concept. What does chemistry mean to you? We're all surrounded by chemicals. I'll later go on to show the synthesis of some molecules that can provide potentially specific diagnoses and really help us understand the way a tumor develops. But remember that water is a chemical. We're all full of water, and essentially we have about 90% or coming on for 100 molar protons in the body. Therefore, there's a lot of potential for getting signal. Using proton MRI, we can interrogate the nucleus of those protons inside the water, and by exploiting so-called relaxation parameters, T1 and T2, and other functions such as the diffusion of water molecules or flow in the blood, we can create pictures like this. This is looking inside the brain, totally non-invasive. It's so common in clinical practice now that I downloaded this from Wikipedia. We can readily achieve resolutions of sub-millimeter, perhaps 250 micron or better, perhaps 2 millimeter thick slices. But this would be normal anatomy. What might a tumor look like? This was a patient who unfortunately had a recurrent astrocytoma. A very poor prognosis, will probably be dead in 6 months. But if you look at this MRI, well, you can see multiple slices, each 2 or perhaps 5 millimeters thick through the brain. We can see some anatomy, we can see some white and gray matter, and we can see some ventricles here of the brain, and we can see distortion. But the tumor isn't obvious. And so what can we do to enhance that? And that's where chemists can immediately move in, not just examining the water, but perhaps introducing a molecular imaging, a contrast agent, to help the tumor show up. If we look at the second row, it's the same patient after administration of contrast agent. Now here we have something called gadolinium DTPA. It's essentially a lanthanide, a gadolinium ion, and the beauty of this is it's paramagnetic. So magnetic resonance is all about detecting the influence of magnets on molecules. And this alters the relaxation properties of any water molecules that are close to it. Now, of course, gadolinium is quite toxic, and so you have to have a ligand around it, which holds it both kinetically and thermodynamically in place so it can't poison the patient. However, a number of these molecules are now available and are in routine clinical practice. What's critical, though, is there's at least one location where water molecules can approach the gadolinium and cause relaxation. Therefore, wherever this molecule goes, you will see contrast. In this cartoon here, what you see is a blood vessel. In the normal brain, blood vessels have a very tight walls. Nothing leaks out of them. But in the presence of tumors, you'll often find these so-called fenestrations where small molecules like this can leak out. And now if we look here, we can see the white signal, the bright contrast, clearly indicating the extent of the tumor. This can then critically help the oncologist, the radiation oncologist, target therapy to try and help this patient. What else can we ask about a patient? Water is endogenous. The contrast agents require infusion. But remember, your body, your cells are made up of many other molecules. And some of these can also be used in diagnosis and prognosis. So this is, again, advanced brain tumor. And one of the things we find is we can do NMR spectroscopy. So this isn't the water now. These are the small molecules at millimolar concentrations that exist within the cells. And these are a number of spectra obtained from different locations through the brain. One of the things about MRI is it can be intrinsically three-dimensional. And so here you see different orientations. This is the brain at one angle. We can cut a slice through where it's yellow and see the other angle. Looking at the spectra here, each of these molecules has a unique chemical shift. The signal along here shows, for instance, at about 3.5 ppm, there's a signal due to choline. Choline is involved in cell membranes and cell development. Tumors that are growing and very aggressive tend to have an elevated choline signal. Likewise, because tumors are growing very rapidly, they tend to lack a good blood supply. So instead of having oxidative phosphorylation, they'll often have a glycolytic metabolism with a buildup of lactate. We can identify lactate at about 1.3 ppm, and then two other signals here, creatine and N-acetyl-aspartate. Healthy brain tissue normally has lots of creatine and NAA, and in fact, if we look at these spectra at the back here, we see those signals. Tumors will often have a good deal of lactate and choline. And so now, simply interrogating other molecules that are already within the brain and the tumor, we can get an enhanced diagnosis. And the reason that this is so critical is how do we know just from these signals that this is a tumor? It turns out, following radiation or following resection of a brain tumor, you will often get necrosis. You'll often get scar tissue, perhaps radiation necrosis. And 6 months after a patient has had the primary surgery, you want to know, is an aggressive tumor coming back that needs further chemotherapy, or do they simply have scar tissue and they will go on to lead a reasonable life? And so this kind of procedure where here we can see lactate and choline coincide with the regions where creatine and N-acetyl-aspartate are missing. So we can interrogate certain molecules, but obviously there are many molecules if you go to biochemistry within the body. How many of them can we detect? And the problem is that we have that 110 molar protons. Therefore, we have to suppress the water, or we can interrogate the water. And obviously, I showed you the gadolinium DTPA, which was used to detect that first brain tumor. And one of the things you'll find is wherever there's a good idea, many manufacturers will jump on the bandwagon, and there are multiple of small gadolinium-based paramagnetic contrast agents. Essentially, they all operate in the same way. There's very little difference between them, but it means multiple manufacturers have a share of the market. What you'll notice in every case is gadolinium tied by this ligand with excess of water. So that's the brain. Much of our research is actually devoted to prostate cancer, and particularly more recently to breast cancer, and that's where we've made the greatest progress in terms of translating our preclinical, chemical, and animal studies, which I'll tell you about later, to the clinic. But for breast cancer, there's actually a good deal of hope, and we'll see, obviously, the common press is interested in this. One of the things we find is a primary issue is detection. Many women will unfortunately develop breast cancer, and it appears that the sooner it's detected, the better the chance of cure, essentially a lumpectomy and removal. But if the tumor is large or has already spread, one may well need chemotherapy. And then there may be the question of what is the right therapy? Can one tell anything about that tumor? So not just its location, but how will it respond to a given therapy? And how can we really tailor that therapy to the patient? And a classic drug here is Herceptin. It's a very fine antibody that will kill certain tumor cells. Unfortunately, only about 30% of tumors or patients express that receptor. So if you give this drug to the other 60% of women, it will have absolutely no effect. But if you can identify those who will benefit, this is very effective at eradicating cancer. What can we do for the other 60 or 70% of patients? Well, as I've said, the first thing is to detect the tumor. So here, earlier I showed you the brain. This is now a breast. And from the MRI, you can see heterogeneity. But what is actually tumor? One of the things you find about the human breast is there's a good deal of fat in it as well. And so if you're doing proton MRI of water, this can appear as dark signal. So this is non-contrast enhanced. We put in this contrast agent, gadolinium DTPA, and again, it leaks out of that fenestrated vasculature of the tumor, helping the radiologist detect the tumor. That's simply a static image. We look at before and after, and we see a change in signal, which can very easily help you pinpoint the tumor. But can we learn more from that? Remember, this contrast agent is administered intravenously. It will travel through the blood. It will leak out in the tumor. And therefore, potentially, if we use rapid imaging, we can obtain contrast curves like this. I showed the signal increased before and after, and here we can actually look at the kinetics. And it turns out that the shape and intensity of this change can be indicative of the aggressiveness of a tumor, which may give a physician the idea as to what kind of therapy will be required. We've actually been applying this in a clinical trial. This is a new clinical trial sponsored by the Komen Foundation with colleagues at UT Southwestern. And the first thing is these women have locally advanced breast cancer, and they're undergoing chemotherapy prior to surgical resection. They're receiving cyclophosphamide and adriamycin. This is standard state-of-the-art therapy. But how can we tell which patients will benefit? It's well known, again, that about 30% of patients have a very good response, but many others are less fortunate. We can infuse this gadolinium DTPA again. We look at these contrast curves, and here's one patient with a tumor that lights up very nicely before therapy. And after chemotherapy, after 4 cycles of chemotherapy, this tumor has totally gone. And in fact, upon resection, upon pathology, there's no evidence for residual tumor. So this is a very good outcome. But we have a second patient here. Well, the tumor appears somewhat larger, but realistically, the contrast curves are very similar under baseline conditions. Upon surgery, this patient still has a very extensive tumor, and in fact, if anything, it's grown. One of the things that could be critical is to intercept this patient very soon into therapy, well, either before therapy to predict how they will respond, or very soon into therapy to see how they're responding, and potentially add additional or different chemotherapy to ensure that they'll have a good outcome, and that's our goal. But if you look at this gadolinium DTPA, we really can't tell the difference in the beginning to predict how the patient will respond. But recently, we've been applying Another challenge. And again here, we're simply turning to nature as opposed to the chemistry laboratory, and we're using oxygen as our contrast agent. Your blood cells are full of hemoglobin, somewhat complex tetramer. And when it binds oxygen, it goes from being paramagnetic to diamagnetic. Now, paramagnetic is just like that gadolinium DTPA contrast agent, and therefore we might expect that deoxygenated blood would yield contrast in an MRI scan. Therefore, if a patient goes from breathing air to oxygen, we may see changes in the signal in their tissues or tumors. And what's quite remarkable here is we look at, this is the same patient I showed you earlier with the gadolinium DTPA. So again, the tumor has gone away, but pre-chemotherapy, in this case, this patient, upon breathing air, had a 20% change in the signal in their tumor. So a very remarkable change, and this is simply from the patient breathing oxygen. The second patient had essentially a very small change, only about 3%. What was critical is, and we've now seen this in 3 out of our 10 patients, the 3 who have the big oxygen response also have a good therapeutic response. We currently have the funding to look at another 10 patients, and hopefully the further data will bear it out and we'll go on for a large clinical trial. Obviously, there's also always the danger of raising the hopes of people inappropriately, because this may just be chance. However, there is a certain logic that a tumor that shows a bigger response to breathing oxygen may well have better blood delivery, which could also be associated with better delivery of the chemotherapeutic agent. But obviously, oxygen as a contrast agent, and this bold blood oxygen level dependent contrast, is telling us something that the gadolinium DTPA isn't. What else can we look at? So that's MRI. And first, I'm just going to review principles that are in the clinic that are used routinely, such as the gadolinium DTPA. Then there were studies such as the BOLD, which are experimental, but they're in the clinic. And later I'll go on to our research. So another instrument that has really entered clinical practice within the past 5 years, although it was developed some 30 years ago, is positron emission tomography. The size of the instrument is very similar, and from the outside you really can't tell the difference. But this looks at radioactivity. It detects gamma rays following positron decay. And the basis here for the clinical practice currently is that we use glucose. And as I was saying, tumors often lack oxygen and are highly glycolytic. They very avidly suck up and absorb any glucose from the blood. Therefore, if you give extra glucose, you may expect it to end up in the tumor. And if you have radio-labeled glucose, that should allow you to detect the tumor. Now, one issue here is that this contrast agent has a half-life of 110 minutes. It's highly radioactive. 110 minutes after you've made it in your cyclotron, half of it's disappeared. Now, that has the advantage that you can give it to a patient, and they can go home, and a few hours later, they're no longer radioactive. Against that, it means you have to manufacture it that morning, you have to use it that day, and you have to make it again the next day. So we have a facility in Dallas, just close to UT Southwestern, that supplies a large part of Texas. There's another one in Houston. They use FedEx a lot to deliver this each morning to the clinics. Just like glucose, it's taken up into the cells, it's phosphorylated, but now, because it's fluorodeoxy, it's no longer susceptible to enzyme activity, so it's trapped within cells. And the beauty of this, and if we're lucky, we'll be able to get this to rotate. Could you just click on it, and it may rotate? The bottom line here, though, is that all these dark spots indicate uptake of fluorodeoxyglucose. The brain was active, so the patient was thinking. You also often see a good deal of uptake in muscle. So in fact, you administer this to a patient, and normally you do the PET exam about an hour later after it's had the chance to be taken up into tumors. And so often you'll see a good deal of uptake in the heart, perhaps the liver. And if they're not immobile, you'll often see a good deal of uptake in muscle as well. Also clearance in the bladder. But you see all sorts of other hotspots. And what this tells you is that this lung cancer patient has a very extensive metastasis. There would be no point in the surgeon resecting the primary lung tumor, because the patient would die of the other tumors. Therefore, what you need is systemic, extensive chemotherapy. If you're lucky, you can start to eradicate the metastases, and perhaps then it's worth going in and tackling the primary tumor. So this can really perhaps save the patient from unnecessary, costly, and damaging surgery when perhaps they wouldn't be expected to survive anyway. But what you know is you need aggressive chemotherapy to hit the metastases prior to tackling the primary tumor. This technique is particularly used very extensively to examine patients who've had a tumor previously to see if any small metastases are coming back. Although the spatial resolution here is only about 5 millimeters, you can detect nodules very early. So that's fluorine 18. And one of the things you'll find is there are actually many pharmaceuticals that have fluorine. Now, the stable isotope is fluorine 19, and we can use that for NMR spectroscopy. And I'm going to be talking about a number of molecules we're developing to exploit the properties of fluorine 19 in a few minutes. In the meantime, just note there are antifungals, there are psychiatric drugs, there are chemotherapeutics, each of which have this fluorine atom. And the beauty of any kind of fluorine spectroscopy is there's essentially no background in the body. So whereas if you want to interrogate millimolar concentrations of lactate, of a drug, of glucose by proton MRI, you have to suppress water. For fluorine MR, we don't have that problem. So there are many potential drugs we could examine to look at the uptake and clearance. But I'll afterwards talk about reporter molecules. So let me just review so far. Currently, radiology essentially identifies the location, size, and stage of a tumor. It's commonly used in breast cancer, brain tumors, as we've seen. A recent innovation was to combine an anatomic procedure with PET. So we said that PET could identify a tumor. But realistically, that rotating image I showed you earlier, it wasn't very clear where the spots were. Which organ are they in? Where does the surgeon go? And so by combining it with a PET-CT, a somewhat more sophisticated instrument, we can have the anatomy, we can have the PET, we can overlay it and determine exactly where the tumors are, which is very helpful to surgeons. This is the state of the art today, but where can we go? What's the prognosis? Now, I showed you that initial study we're doing in the breast cancer patients breathing oxygen to try and get a handle on how they respond to chemotherapy, and now I'll talk about some other agents we're developing. Particularly, how might one characterize a tumor? One aspect would be physiology. How much oxygen is in there? Hypoxic tumors do not do well. What's the pH? What's the blood flow? There are many parameters one could examine. What receptors might there be for drugs, like that Herceptin? How might one examine gene expression to understand what kind of gene therapy one could use? And oxygen is particularly important because hypoxic tumors, those that lack oxygen, do not respond well to radiotherapy, and they tend to be much more aggressive. Now, in the past, while tumors hypoxic, what can you do? But today, there are alternatives. You can give a more intense dose of radiation in the hope of eradicating that tumor that's hypoxic and resistant. You can use a hypoxia-selective cytotoxin drug, such as tirapazamine, which specifically kills hypoxic tumors. And we have this kind of Accuray robotic arm radiation delivery device now, so that if we know that a part of a tumor is hypoxic, we can deliver a higher radiation dose to that tumor. So just to remind you, it's been known probably about 50 years now, but this was from a review, that hypoxic tumors, tumor cells, tend to resist radiotherapy. And this is because in the presence of radiation, you get free radicals on the DNA. This can react with oxygen to form peroxides, which can cause all sorts of damage to the nucleus of the cell. In the absence of oxygen, you can have repair here, and hence chemical restitution and recovery of the cells and resistance. So given that hypoxia is important, there are many molecules that have been developed to examine this. You'll see a common thread here that every one of these molecules has a nitroimidazole ring. They also all have a side chain here, and in every case here, you'll see a fluorine. Now, the basis for these molecules is that in cells, nitroreductases will reduce them, forming variously reactive radicals and ultimately reactive amines. In the presence of oxygen, they'll be reoxidized. So these can be administered intravenously. They'll go to a tumor. They'll be subjected to these nitroreductases, but in the presence of oxygen, they'll clear out again. They'll wash out and they won't accumulate. But in the absence of oxygen, they'll accumulate. If we then put fluorine 18 here, just like the fluorodeoxyglucose, we can detect it by positron emission tomography or potentially by NMR. Also notice here we have a single fluorine. Here we have 3 fluorines. Here we have 6 fluorines. By the symmetry, by the nature of the structure, here we would expect to have 6 times more signal. Therefore, for the same amount of interrogating reporter molecule, we should get a much better signal. However, those molecules are required at millimolar concentrations. They tend to be quite toxic, and they're very difficult to detect. And so we've actually chosen another approach, which is based not so much on the biochemistry and the molecular damage to the DNA, but rather on directly detecting oxygen based on physical principles. So oxygen dissolves in perfluorocarbons. This is a prototype molecule, hexafluorobenzene, which we like to use. It has 6-fold symmetry, so it gives a single NMR resonance. We can inject as little as 50 microliters, which is really less than a drop, into a tumor and make multiple measurements as to the absolute oxygen. Not just some idea of hypoxia, but actually measure the PO2. We're fortunate there's this relationship, a linear relationship between the spin lattice relaxation rate and the PO2, and it has a single signal. If you want to know more about how this works, you can turn to Henry's law and the various other ideal gas and liquid laws in your textbook. In the meantime, though, we have a reporter molecule. Somehow we have to get it into the tissue. We have a very mobile liquid. We can use a thin syringe. Here we see a penny. It's a 32-gauge syringe. We can deposit it into various parts of a tumor. We can do proton MRI to show there's a tumor. We can do fluorine MRI to see what we're looking at. And then we can do oximetry with some clever spin physics. And I don't know if it should be called spin physics or spin physical chemistry, by which we can get the amplitude of the signal, and by varying a specific delay time here, we can get a relaxation curve which directly gives us oxygen tension. So now, at somewhere like 50 to 150 individual locations with a tumor, we can simultaneously measure how much oxygen there is. There are clinical trials going on right now where this is done with needle electrodes. You take the needle, you stick it into the tumor, you make a measurement. You move it forward, you make a measurement. You move it forward, you make a measurement. 150 locations, and it correlates with clinical outcome, particularly for tumors of the head and neck, and in some cases of the cervix. But all you can do is make one measurement, and it's highly invasive. Here, the beauty of it is we can deliver the reporter molecule, and we can make multiple measurements, and we're making all these measurements simultaneously. So in this case, we had a tumor implanted on a rat. The work I'm talking about now is preclinical. These are investigations that we hope to translate to the clinic eventually. And we can make multiple measurements simultaneously, and we see that within this tumor, some parts are essentially hypoxic, essentially no oxygen. Some parts have somewhat more oxygen. We can then allow the animal to go from breathing air to oxygen, and intriguingly, those areas that started better oxygenated go up, and the others don't respond. We can switch to an alternate gas, so-called carbogen, which has 5% carbon dioxide, which is thought to be vasoactive and modulate blood pressure, and we see essentially the same effect. So what we know is if we irradiated this tumor, there might be better response here, but much of the tumor would have no response to breathing the elevated gas. However, we take a different kind of tumor, a so-called HI tumor. It grows more slowly, has better vascular development, and now, if we go from breathing air to oxygen, we see that although under baseline conditions, essentially the whole tumor is rather hypoxic, with oxygen, essentially the whole thing is oxygenated. So we've got oxygen throughout this tumor, and we can make that measurement. And when we irradiate these tumors, this would be a control tumor that doubles in size after 20 days. If it's irradiated while breathing air, you can see some growth delay. If it's irradiated while breathing oxygen, you can see a much greater growth delay. Essentially, these animals lived 4 times as long with their tumor because the radiotherapy was far more effective. One of the things you need to remember is that most tumors in patients grow much more slowly than in animal models. Part of this is the nature of the models we have, and part of it is the impatience, the investigators who want to see a rapid result. But our suggestion is that if a patient had a tumor like this, by identifying that we can modulate the oxygenation, it would be worth that patient breathing oxygen during radiotherapy because we'd expect a much better clinical outcome. And what we can see is that in this kind of tumor, initially, something like 60 or 70% of it is rather hypoxic, and we can wipe that out. And so our goal is to move this in the near term from animal investigations to the clinic. However, that is fluorine MRI. And I was just mentioning that currently we have a 1.5 Tesla clinical system, and we will get 3 and 7 Tesla. Currently, we cannot do fluorine MRI in the clinic. With the new systems, we should be able to. But we were impatient in terms of moving this to the clinic, so rather than waiting for the new MR system, waiting on the physicists and the engineers, we turned to our chemistry catalogs. And we said, what is it about hexafluorobenzene that allows us to interrogate oxygen? And there were a number of characteristics: the symmetry, the lipophilicity, the mobility, and the inertness. And it occurred to us that hexamethyl disiloxane has many similar characteristics. You'll see it's an entirely symmetrical molecule. It has one kind of proton. And because it's a methylsilane, the chemical shift is maximally removed from the water and fat signals. Therefore, we actually have a chance of detecting it in the presence of water. And we know we can do proton MRI. We know we can detect small signals, such as that choline and creatine and lactate we saw earlier. So perhaps we should be able to detect this by applying appropriate pulse sequences to eliminate fat, eliminate water. And again, what's important is we find that the spin lattice relaxation time is directly proportional to the oxygen tension. And here in a phantom, you can see that we have water in this cylinder, fat in that cylinder, and hexamethyl disiloxane here. By applying our spin physics tricks, we can see just the signal we're interested in. And here we're looking at it in the thigh of a rat as the initial model. Here we can detect where we've put our reporter molecule. Here you can see without water suppression, we have water, fat, and hexamethyl disiloxane. We apply the spin physics just to our molecule. We can interrogate the spin lattice relaxation time. We can determine the PO2, and we can look at changes in going from air to oxygen to air. This is something we just discovered in the past year. And so now we actually have our own competing technologies, and we'll see which one moves forward fastest. What is most critical, though, is hexamethyl disiloxane has an extensive literature showing that it's non-toxic, and therefore we should be able to apply this to developing studies. But of course, we have to inject this into a tumor. And it's never a pleasant sight, but when we're working on cancer, we need to remember that we're dealing ultimately with patients' lives. And so this is a patient who has a head and neck mass. It's ideally accessible for radiotherapy, and it's ideally accessible for putting in these kind of reporter molecules with a small needle so we can make measurements. This might be less appropriate in a brain tumor. So, okay, we're developing some methods, at least in animals right now, and ultimately they may be developed for the clinic to look at oxygen tension. But what can we look at next? And next I'd like to turn to receptor expression. Because it turns out that there's a molecule, phosphatidylserine, which exists in the lipid membrane bilayer of all cells. And normally, there are enzymes, scramblases and flipases, that ensure that this lipid all faces onto the inside of a cell. But under stress conditions, those enzymes fail, and some of it is exposed on the outside of the cells. And not just on the outside of the cells, but in fact on the outside of the endothelial cells lining the blood vessels. Therefore, we may be able to detect this molecule in the body. And in fact, here we have antibodies designed to detect phosphatidylserine, and you can see in the histological slice the extent of it, anti-CD31, which detects blood vessels, and you can see they're essentially coincident. And the beauty of this target, then, for tackling tumors, is it's directly accessible to the blood. Because whenever you have a drug, how will you get it to a tumor? If it's inside the blood vessels, you've got direct access. And just going back to the chemistry, this molecule already goes by a number of acronyms that we use. When my colleague Phil Thorpe first discovered it, it was called 3G4. Then he humanized it, so it became chimeric 3G4, or has a USAN name of vatuximab, and they're hoping to develop a trademark of Tarvacin, and this is entering clinical trials. You can also see, if you go to SciScholar, Chemical Abstracts, the simple target, phosphatidylserine, comes by many names, which you can read and indulge if you like. So we have an antibody, but what can we do with it? We know, I showed you a histological slice, it can target a blood vessel in a tumor. So that may mean it could be a therapeutic, but could it also help us detect tumors? And so we've actually just been funded by the Department of Defense Breast Cancer Initiative to try and develop this as an imaging agent to detect tumors, to detect the expression of phosphatidylserine, and to combine it with radioimmunotherapy. And for this, we've turned to another branch of chemistry, so radiochemistry, and specifically arsenic. Now, when you hear arsenic, you may well think of arsenic and old lace and poisons and poisoned groundwater, and it's not very good news. However, it's monitored at parts per billion where it's toxic. When we're using positron emission tomography, we can be at least 1,000-fold less in concentration. And so wherever you have a poison, just consider if you have the appropriate concentration, it may still be useful for something. Now, arsenic has a number of radioactive isotopes, and you can see that the half-life ranges from 1 day to 80 days. These isotopes are radioactive. They can be a little difficult to get hold of. Currently, our only supply of arsenic-74 is from Brussels in Belgium, so it has to go on a truck, go on a plane, and arrive to us, and negotiate Homeland Security and customs, and sometimes it's sat at the airport for a while, but we get it, and because it has such a long half-life, it can still be useful for us to do investigations. We're also obtaining arsenic-77 from the nuclear reactor at UT Austin. You can see it's got a much shorter half-life, but it doesn't have to travel so far. And some of these emit positrons. So just in the way that I showed you, fluorodeoxyglucose could be used in positron emission. earlier, so could these ultimately be used for detection. However, arsenic-77 can be used as a therapeutic. It emits electrons which can cause local tissue damage and therefore might be a therapeutic. But of course, now we have our arsenic and we have our antibody, and how do we bring them together? And here again, there's a role for the chemist. We have our antibody. We can take this small SATA derivatizing ligand, and under appropriate chemical conditions, we can expose a thiol group, which will react with arsenic triiodide, which can be oxidized, and this is stable. There are a number of things that are particularly critical here. Why do we simply not react this with the thiol groups that occur naturally in antibodies? Well, they're usually involved in the tertiary structure of the antibody, and if we start reacting with those, we may well perturb the structure and no longer have the activity in targeting the receptor we're interested in. Moreover, here we can much better control how many arsenics will be on each antibody, and certainly we find that after derivatization, this antibody retains its targeting activity. We can again evaluate it in animals. All these investigations require preclinical tests. Here we have a rat with a tumor growing in the thigh. And this is simply phosphoplate planar scintigraphy. So the animal has been dosed with a radioactive antibody, and every few hours we can simply lay it on a phosphoplate detector and detect the amount of signal. And here we can see changes in signal intensity. Here you can see the image with accumulation after about 48 hours that's particularly high in the tumor. And one of the things this tells you, which is very common for antibodies, is their biodistribution, their pharmacokinetics can be very slow. So the optimal target to background ratio may not be achieved until 48 or 72 hours. And you can imagine if you were trying to use fluorine as the PET reporter molecule, as we did with FDG earlier, where you have a 110-minute half-life, you'll have essentially nothing left after 24, 48, 72 hours. Whereas with this isotope, we can really look at the biodistribution. We're looking at concentrations here that are probably picomolar or less. You essentially can't see them in your test tube prior to injection. You can only detect them based on the radioactivity. But we can again do the PET, and this is now overlaid on the MRI. So the MRI shows us the anatomy, and the PET shows the distribution of the agent within the tumor. So potentially we feel this method could be used to detect tumors or to detect expression of the receptor, telling us that therapy based on the antibody could be useful. That can have two approaches. At low doses, we could have arsenic-77 attached to it, which causes irradiation of the tumor and damage. Or at higher concentrations, it turns out that the binding of the antibody to the receptor can attract macrophages, lead to thromboses. When you block blood vessels, you cut off the blood supply and kill the tumor. And so we're very hopeful that we'll be able to develop this. Certainly, we've received some funding, and it's also starting to enter clinical trials. So that's the second aspect. A third aspect that we're working on is gene expression. It turns out that tumors often have deficiencies in genes. They may have an overexpression or an underexpression. We may want to use gene therapy to modify their behavior. Or in another case, there are a number of drugs, for instance, 5-fluorouracil, which I showed earlier, that are incredibly toxic. If you give them at the wrong dose, your patient may die. However, if you give 5-fluorocytosine as a prodrug, it will be metabolized by the enzyme to 5-fluorouracil at the location of the enzyme, and that's highly toxic. Therefore, you can systemically deliver a relatively non-toxic drug which becomes active in the tumor if you can have expression there. So there are many reasons for knowing how do you get 5-cytosine deaminase into a tumor. Somehow you have to deliver it. And so whenever we're trying gene therapy, there are a number of important aspects. One, if we deliver, let's say, an adenovirus, which can transport our therapeutic gene to the tumor, does it ever reach its target? And if it does reach the target, is it uniform or just in one location or many? Is it expressed? We may be administering a bacterial, a plant, a yeast gene. Will it be expressed in a mammal or man? And for how long? One of the things you may have heard about was in Philadelphia a few years ago. A young man was receiving gene therapy. He got a viral overload, stress to the liver, and he died. And so there can be toxicity. So certainly, you may want to administer this once, but not necessarily over and over again. So you want to know how long your expression is going on. Now, your therapeutic gene may not be directly detectable. And so a new trick that's become very popular in the past few years is to use a reporter gene in tandem. And then you know that if this reporter gene, which you can detect, is seen, your other gene must be present as well. And there are a number of possibilities here. There's green fluorescent protein. If you think of jellyfish, they can fluoresce. And essentially that gene can be transfected into tumor cells. Now, this may not be immediately clinically relevant because light doesn't travel through the body very well, but this can be very useful for preclinical studies in mice. There can also be luciferin. If you think of the firefly, it glows, and this gene can be transfected into mice. We can also use various radioactive labels and beta-galactosidase, which I'll talk about in a few minutes. But so here we have brain tumor cells that were transfected to express green fluorescent protein. When we place the mouse under a fluorescent camera, these tumors glow green, so we know where the tumor is. And so if we were looking at a therapy and these tumors started to spread, we could detect that without having to cut the animal open, which is very important. It saves the number of animals, it saves the cost, and often drugs, particularly at the preclinical stage, are in very short supply. Chemists don't normally optimize the manufacture of a drug until it's needed in large quantities. Here's the luciferin growing in another tumor. Now, how does this luciferin work? So essentially, luciferase, the gene, is transfected into the tumor cells, which are then implanted into the animal. You'll see these tend to be nude mice because we're trying to grow a human tumor in a mouse, and normally the immune system would prevent the growth. However, these nude mice are so-called athymic and have no immune system and a very nice model for the growth of human tumors. Something else to bear in mind is that although when you see a firefly flashing in the garden on a warm evening, it's very bright, by the time it's in an animal, the signal is actually very weak. And therefore, we need a black box with a very sensitive CCD camera. The chemical reaction essentially just needs some oxygen, some ATP, and D-luciferin. And we have to buy the D-luciferin and inject it essentially anywhere we like into the animal, and very soon a tumor will glow. Intriguingly, and the reaction has been studied quite extensively, depending on the pH of the location of that enzyme, one may get essentially green light or red light. And we're essentially using the luciferase gene, but there may be reasons to use other genes as well, for instance from jellyfish, using things like cilantrazine. So there can be various molecules, some of which are much more complicated than others. And whereas the bug synthesizes this very efficiently, it's actually quite complicated to synthesize in the lab, and it's not cheap. But let me show you an application. So here we're testing a chemotherapeutic. A very popular clinical chemotherapy is based on cisplatin. But how effective is it? And might one need to combine it with other drugs? And so here you can see we've implanted tumor cells that express luciferase into an animal, and under normal conditions, the tumor will simply grow and the light gets brighter and brighter. If we give cisplatin as the chemotherapy, you see the tumor goes away. Therefore, we can non-invasively in a single animal evaluate the efficacy of this drug. And we're looking at many drug combinations, because one of the problems is, although this tumor has gone away here, it may come back again. In fact, if we look at this animal a week later, light comes back again. And that's when we start administering secondary drugs. But again, the whole thing is non-invasive. We've actually been using this luciferin approach to tackle another drug. This is an experimental drug in phase 2 clinical trials, cumbretastatin, made by Oxygene. It was extracted from the African bush willow, and it's a vascular targeting agent. Where it finds immature blood vessels, it binds to tubulin and causes vascular collapse, again starving the tumor of oxygen and nutrients, and should be very effective as a therapeutic. And so here we have a tumor growing in a mouse. We inject luciferin, and we see the signal go up, because obviously the drug, our reporter molecule, has to go through the body, through the blood, to the tumor, generate the light, and progressively it's consumed, and so the light comes down again. If we inject saline, so sodium chloride solution, afterwards, and 2 hours later repeat this, we see essentially the same pattern. However, in this animal, we first looked at the control, we then injected cumbretastatin, and 2 hours later you see there's much less light. In fact, if we compare it, we see that the intensity of the light has gone down some 70%, and the time to peak has gone up by many minutes. And so this now is another way of looking at the activity of a drug. We know we've shut down the blood supply. You may ask why we need so many techniques. Well, one is that an MRI system can easily be $1 million. You can buy a system like this from Xenogen for about $250,000, or you can build it for yourself for a few $1,000s, which is what we've done. But that was non-invasive. How do we know that what we think has happened in that tissue has really happened? And again, here's another opportunity to explore chemistry. Many of these Hux dyes were generated in the 1800s. The German pharmaceutical industry was very effective at generating dyes that would bind to specific pathological situations in tissues, perhaps into lipid or other situations. So here we have a perfusion marker, this Hux dye. You'll see a polyaromatic ring which fluoresces blue. We can infuse that into an animal. We can then kill the animal, we can cut out the tumor, and we can see how many of the blood vessels are perfused, and that will tell us the patency of the vasculature. We can also use an antibody that specifically hits blood vessels. However, how do we observe the antibody? In this case, with the cyanin dye. And here again, there's been a challenge for chemists, particularly Alan Wagner at Pittsburgh, would put in extra double bonds here, and he could change the wavelength. So you'd have Psi3 here for 3 double bonds, or Psi5 for 5 double bonds, and he could change the optical characteristics. But using this in the histological slice, we can see where the blood vessels are, and if we overlay it, we see that many of the blood vessels have blood going through. 2 hours after cumbratostatin, we see there are still blood vessels in another tumor, but now there's no perfusion. So this now provides histological evidence proving that what we'd seen non-invasively is valid. And this is very important, because when you're doing it non-invasively, there's a leap of faith as to whether what you're seeing is really there. However, here, this required 2 animals. When you looked at the bioluminescent or the other non-invasive imaging procedures, a single animal gives you the whole set of results. But something else to remember is that animals are not 2-dimensional. That was a planar image. But in fact, we can do this in 3 dimensions. So we can have a mouse, and if we put it between multiple cameras, we can take an image from multiple sides. And you may never have thought about this, but light will penetrate through tissue. It scatters a lot, and there's some absorption, but some will come out. And so in this case, we have A549 human lung tumor cells. An interesting model, if you inject lung tumor cells into the tail vein of a mouse, they will colonize the lungs and grow. And this is a very nice model for lung cancer. Here we can see extensive colonization of the lungs. If we look from the backside, there's much less light coming through because it has to go through more tissue. Multiple angles, if we do appropriate mathematical convolution and modeling of the light that comes out on each side, we can make a 3-dimensional representation. So here we see multiple nodules throughout the lobes of the lungs. Here we can overlay it on the animal to give the anatomical orientation. Something else that's interesting is often when you inject these cells, some of them will also grow at other locations. And so this is obviously somewhere down near the tail. This was obtained non-invasively. This animal could have lived on. We actually sacrificed it, and you'll see there's already very extensive colonization of lungs here. But the beauty of these procedures is non-invasively, we can look at how a tumor develops. So that's luciferase, luciferin, and really just in the last 5 years, it's taken off as a very popular tool for biomedicine, for looking at tumor development and many other aspects of molecular biology. But prior to that, there was beta-galactosidase. In fact, this led to the Nobel Prize some 50 years ago, and if you go to Medline, you'll see some 14,000 citations. And therefore, it occurred to us, if we could interrogate what beta-galactosidase is doing, our reporter molecules could be much more versatile. One of the other things that's interesting is, if you take luciferase, your luciferin molecule has to have a very specific structure. If you take beta-galactosidase, you can go to catalogs and buy all these different structures. Each of them will give a different color, and each of them show activity with the enzyme. In fact, there was even one reported for MRI activity from Tom Mead, but it's a rather complicated structure, and it doesn't enter cells. And how do you detect enzyme activity if your molecule doesn't enter cells? So in fact, what he required was an intracellular direct injection using a microneedle, and then he could detect activity. But that's not necessarily useful for tumors. But it occurred to us as a first opportunity that we could simply add a fluorine atom to one of the traditional so-called yellow biochemical indicators. You can see the structure here, and by being para to the glycosidic bond, we expect to see a signal. So my chemist was able to synthesize this, and we see a single signal. We add the enzyme beta-galactosidase, and we see liberation. It turns yellow, and a chemical shift. And wherever there's a chemical shift in fluorine NMR, you can start to learn something about the biology. But that was para. Obviously, as chemists, we love to make isomers of molecules to look at differential activity. So we can put the fluorine in the ortho position, perhaps move the nitro group. Now we can have multiple isomers. And fluorine NMR is incredibly sensitive to the local microenvironment. And so here we see each of these molecules, although chemically really quite similar, has a different chemical shift. We can look at 3 substrates simultaneously. We can look, upon addition of the enzyme, at the conversion to 3 products simultaneously. And one of our goals here is that potentially we could make substrate sensitive to 3 different enzymes and therefore start to interrogate biochemistry non-invasively. Here we just added the enzyme, but we've also simply added this to cells or used it in animals and seen activity as well. Isomers, we might expect different biochemical activity, and indeed, some of these react much faster than others. And what we found was if we look at the pKa of the product, there's a linear relationship with the rate of reaction. Now, I've said that the enzyme is very non-specific, very promiscuous, but in fact, if we go to the alpha anima, there is no activity. So there is some degree of specificity. And this has been reported very extensively in the literature for optical photometric approaches, but we're using it now for fluorine NMR. And here we have a prostate tumor expressing beta-galactosidase implanted in a nude mouse. We inject some of this material directly into the tumor, and we see over a period of 17 minutes conversion. Therefore, we know that that tumor is active. Again, this is a very early stage of development, but we're hoping that down the road this will be useful for detecting gene expression and gene therapy. Our signals are also well enough resolved. My talk is meant to be about imaging rather than just spectroscopy, but what is spectroscopy? It's simply localized imaging. So here, we can do selective imaging and determine each of these signals separately. So if we want to, we can look at substrate and product in each of 2 vials. This would be a non-selective image. One vial has one has the other. We can detect them simultaneously. So rather than just doing a global spectrum down the road, we should be able to detect where we're looking at within a tumor. Again, one of the things I mentioned, and people in Las Vegas appreciate this, every spin wins. And NMR is all about spin physics, how many nuclei you have. And I think you noticed earlier on those hypoxia molecules, we went from 1 to 3 to 6 fluorines. Likewise here for our reporter molecules, we may want to go from a fluorine atom to a trifluoromethyl group. And here we can see we add a trifluoromethyl group onto a very similar substrate, get a chemical shift, we get a conversion. But the beautiful thing here is we needed 3 times less reporter molecule. We can also add all sorts of other reporter molecules on there. But what's next? As I mentioned, there's a problem with fluorine NMR that is not necessarily clinically available. And so wherever we have a fluorine investigation, we try and create a proton analog. In the last few slides, I'll just show you our capability here. So it turns out if you go to the Aldrich chemical catalog, you can buy something called sGal. Again, we have a glycosidic bond here, presence of beta-galactosidase, it releases this tricyclic structure. In the presence of ferric ions, this forms a black precipitate. And it occurred to us that where you have ferric ions and a precipitate, you may well have paramagnetism. So here we have an optical result. These are breast cancer tumor cells in gel. In the presence of beta-galactosidase, sGal, and ferric ammonium citrate, it turns black. If we do the NMR experiment, we see signal loss. That's in the test tube. Here we go to an animal. And it's somewhat subtle in this initial experiment, but if we do subtraction images of an image at 35 minutes versus 5 minutes, we see loss of signal around the periphery of the tumor, which becomes more intense with time, because obviously the enzyme reaction takes time. And if we do the histology, we find the signal around the periphery, but none in the center, because the tumor was necrotic. Perhaps more convincing is this experiment, where here we did an intratumoral injection. This mouse had 2 tumors, a wild-type tumor that doesn't express beta-galactosidase, and one that does. We inject our ferric ammonium citrate and sGal into both tumors, and here we see essentially no change, and here we see signal loss, telling us that tumor is expressing beta-galactosidase, the transgene, and therefore this would help us evaluate gene therapy. And obviously we have the biochemical correlates as well. So biologists love to have acronyms. NMR spectroscopists love to have acronyms as well. So for here, we've come up with MAGIC, or magnetic resonance assays of gene imaging constructs, and we have the 2 approaches. The fluorine approach, simple, sensitive, multiple reporters and multiple parameters potentially. The proton allows us to do the imaging and see heterogeneity, and we were rather pleased to make the front page of Current Medicinal Chemistry. But one of the things I'd like to emphasize, I've tried to give an overview here of chemistry and how it relates to oncology and cancer imaging and how it may be used to enhance and ultimately cure patients. We can look at water molecules, we can look at endogenous metabolites, we can look at drugs, we can look at physiological parameters, simply by choosing or tailoring the particular reporter molecule. But one of the things I think has been really important is, although I trained as a chemist, I don't work with a whole lot of chemists. I have 1 chemist in my team, I have a molecular biologist, I have a physicist, there are computer engineers, there are data analysts. And just as you see at the Bush Library, walls can be broken down. And I think it's really important that although we may be studying chemistry, we interact with people from multiple disciplines so that we can have animal models. This is a human brain tumor. This is an animal brain tumor, spontaneous. It wasn't implanted. The animal just develops it. So we can have imaginative new molecules so that we can take a gene from a firefly and use it for chemistry. There's a whole periodic table out there. There are many things you can look at and consider. As I mentioned, I have quite a sizable team. 5 years ago, I had one postdoc. Now we have 12 investigators in my team. You'll see their names here, particularly Darwin Zhao, a very skilled physician-scientist, Zhanxin Yu, a chemist, Vikram Kodibagkar, a physicist, Li Lu, a molecular biologist. We collaborate with investigators in radiation oncology. And that was just for my immediate lab. When I go to the radiochemical experiments, we're interacting with many other institutions. You can see a number of investigators and institutions here. And so finally, it just leaves me to thank the many sponsors of our research. Not only do we need individuals, but somehow we need funds, particularly the Department of Defense, but also the NCI and American Cancer Society. And finally, with that, I thank you for your attention. I've certainly been excited to see so many of you here.
Thank you, Dr. Mason. Given the hour, I think we'll adjourn. If people have specific questions, we invite you to the front to talk to Dr. Mason personally. Join me in thanking him again.